Hello everyone, and in this series of videos we're going to go over the gastrointestinal system. Uh, Before we get into any pathology, the presentations of the pathologies, how to diagnose and how to treat, I'm going to go ahead and first go over the digestive process, how it normally should function, and some of the absorption process. Won't get into too much on the cholesterol and LDL and HDL after the absorption, or how detailed the endocrine system is. We'll do that in other videos based on nutrition, cardiology, and endocrinology. First thing I want to start off is reviewing the anatomy real quick of the GI. The gastrointestinal system technically begins at the lips and it finishes at the anus. It is approximately 20 to 30 feet long in the average adult. If you're 7 feet tall, it's obviously going to be longer. If you're 4'5", it's obviously going to be shorter. The first thing I want to discuss is the mechanical function of the gastrointestinal system. It works with a series of peristalsis and segmentations that is provoked by the food that you eat, actual stimulation of what is in your stomach and in your gastrointestinal system. And at the same time, it's also provoked by the vagus nerve, okay, which is an outside stimulus coming straight from your brain. The gastrointestinal system does have an enteric nervous system, which is huge. It's huge. You could compare it to that of the brain and the peripheral nervous system. That's how many nerves the gastrointestinal system has. The vagus nerve is an addition. It's a nerve that is connected to your brain, and your brain receives information and sends stimulation via acetylcholine through as the neurotransmitter through the vagus nerve, and it also stimulates your stomach whenever you smell food and you feel hungry. The vagus nerve is the one that makes you begin peristalsis, all right? It makes you begin secreting acids. If you cut that vagus nerve, okay, now we're not talking about cardiac, just strictly gastroenterology, because the vagus nerve also has some function in the cardiac level. If you cut the vagus nerve, your gastrointestinal system will still work because you have your enteric nervous system. And your enteric nervous system will digest food and cause all the enzymatic secretions that you need for digestion and absorption of food. You just won't be able to smell food and stimulate hunger. Okay, so your gastrointestinal system can work without the vagus nerve. Okay, starting at the mouth. We have mouth. You chew whatever food you're eating, and in your mouth you mix this food with saliva, which holds amylase and starts breaking down some of the carbohydrates and some lipase as well. It's not very significant, but the importance of your mouth is that your teeth and the saliva allow to make the food softer, and you break it apart more. So by the time it gets to your stomach and your intestines, the food is in smaller particles, and it's a lot easier for the acids and all the enzymes to break them apart even further. Remember, they have to cross membranes that are very microscopic. So the more you could break this food down, the better it is. So yes, it is good to chew your food before you swallow, okay? The first action is all controlled, okay? It's voluntary. You propel with your tongue the food to the back of your throat, and there you have the beginning of your proximal esophagus, which you do control. As you propel food down your esophagus, it gets to a point where you no longer control it, and now peristalsis takes place. Peristalsis is just a movement of skeletal muscle, and it is stimulated whenever there's something inside of the gastrointestinal system that has some bulk to it. It does stimulate peristalsis. There are two waves of peristalsis that take place in the esophagus. The initial one, when you initially stimulate it with something, and then a secondary one, which clears up any residue that's left over. Okay, think about it like squeezing a brand new toothpaste tube from the very back to the very front, and then doing it a second time to make sure you get that extra little residue that could have been left over. As peristalsis moves down towards the stomach, you join an area where the esophagus joins the stomach. It's called the gastroesophageal sphincter, or the lower esophageal sphincter. And what happens is that there is no valve there, okay? What happens is that there's a difference in pressure and in size. As the peristalsis moves the food, or the bolus, it's now considered a bolus, into your stomach, the increase in pressure dilates that sphincter, that stricture, and it allows food to enter your stomach. Okay, along with swallowing the bolus, you also have some air. Every single day we not only breathe in air, but we swallow air. Some of it comes out as a burp, and some of it goes all the way to your anus and comes out as a fart, mixed with the anaerobic leftovers from bacteria, which forms gas. Okay, so when you pass gas, it is a mixture of gases from bacteria and air that you swallowed. Okay, I wouldn't be able to tell you how much is which, but a significant amount of it is air that you swallow. In your stomach, what's going to happen now is that you're going to have the stimulation both via the vagus nerve and the enteric nervous system, which has just been stimulated by the food that's mechanically, physically there. And you're going to have stimulation of your G cells, which produce gastrin and stimulate mucus and hydrochloric acid. But the important thing about the G cells is that gastrin is increased. All right, and gastrin stimulates the mucosal membrane to protect your stomach lining, the endothelial cells. You're also going to have stimulation of your chief cells, which produce pepsinogen, and in the presence of acidity, they turn into pepsin. This helps break down proteins. Okay, and you're going to have hydrochloric acid, which not only helps break down the hydrogen bonds and other bonds in the protein, but it also helps kill bacteria that you just finished swallowing, because your mouth is full of bacteria. Okay, what happens here is that for the next hour to 4 hours, the stomach little by little starts mixing. It starts shaking up whatever food, whatever bolus you have in the stomach, until it starts getting broken down to anything less than 2 millimeters, or approximately 2 millimeters. Okay, so your pyloric sphincter is approximately 2 millimeters in diameter. So this food gets broken down into the smallest particles possible, but 2 millimeters or less can pass through this area, okay, without being forced, of course. What happens at this time is that you have something called chyme, and it's a mixture of acids, enzymes, and the food that you've just swallowed into your stomach. Your stomach takes anywhere between 2 to 5 hours, so for sake of just having one number, we're going to say that it takes an average of 4 hours for your stomach to empty completely. Okay, but within the first 15 minutes, not even within the first 15 minutes, within the first 5 minutes of you eating something, you're already going to have enough breakdown to go ahead and start filling the duodenum. Okay, so the chyme now passes into your duodenum via the pyloric sphincter, and at this time you're going to start stimulating the duodenum. Okay, there's a bunch of cells in the duodenum. For right now, I'm only going to talk about the S cells in the duodenum, in the crypts of Leverkuhn. Okay, and they release an enzyme called secretin. Okay, an endocrine called secretin. There's also cells called the I cells of the duodenum, and they're going to stimulate a hormone called CCK, cholecystokinin. Okay, so remember that once in the duodenum, food will stimulate the secretion of secretin and the secretion of CCK. CCK enters your bloodstream and eventually makes its way to the gallbladder, okay, the blood vessels that do feed your gallbladder, and that hormone not only stimulates the gallbladder to contract and release whatever bile was in there before, but also whatever it also stimulates the liver to start producing even more bile. Okay, so if you take out the gallbladder and you had a cholecystectomy and you no longer have a gallbladder, CCK won't have a gallbladder to stimulate, but it still stimulates your liver, and your liver does have some storage of bile and will make bile readily from cholesterol and other things that we have in the body. What happens now in the duodenum is that immediately bicarbonate has to be secreted. The importance of secretin is that it stimulates the pancreas to release bicarbonate, and this bicarbonate is very important because if you have a very low pH in the duodenum, it's good in the stomach because you have protection in the stomach from mucous membrane, a very thick mucous membrane compared to that of the duodenum, and it also kills bacteria. But in the duodenum, you have proteins, these enzymes, and they help break apart your carbohydrates, your sugars, your proteins into the simplest form possible. these enzymes cannot work in a low pH environment because there's too many free radicals, there's too many hydrogen particles that activate too much with everything. It doesn't let them function, okay? So you need bicarbonate secretion into the duodenum in order for these enzymes to work. At this time, what happens is that you have fats, okay? And these fats are very long chains with a lot of hydrogen around them. They need to get broken down, okay? They need to get broken down a lot less, to a lot smaller size. And at the same time, they need to become absorbable, and they need to be able to move around plasma in our blood. In order to do that, bile binds to this, okay? You are going to have lipase from the pancreas being released, and lipase is going to help break down these fatty acids into small particles. But bile grabs these small particles after lipase has turned them apart even more, and it breaks them down into something called micelles, which are even smaller. And these micelles are now easily a combination of bile and these fatty acids absorbed into the bloodstream. Okay, it's a passive transport system, and we'll go over that in just a moment. What happens now is that carbohydrates are also broken down. You have sucrase, table sugar, right? Whether it's brown or white sugar, it's sucrase, and it's a combination of fructose and glucose that's broken down by sucrase. Okay, you have lactose, you have a lactase that breaks that down into galactose and glucose. Okay, so the simplest sugars are galactose, glucose, and fructose, and they're readily absorbed passively through the GI. So, but all these enzymes need to work and break all this stuff down. Okay. Now, what happens at this moment is that you have you begin to absorb all these nutrients, essential nutrients, proteins. Okay. You're going to have amylase, and you're going to also have another hormone that's not readily spoken about that much, but it's elastase, and it also breaks down proteins. And now you have amino acids as well. So you're going to have fatty acids, you're going to have the simplest forms of sugar, and you're going to have these amino acids, which can now more easily transfer through the cells of the lumen of the duodenum and into our circulatory system, okay? And in the case of the fats, which were micelles, they now become, in the interstitial space and in cells, they become something called a chylomicron when they become attached to something called lipoproteins, okay? And that's where the LDLs, apoprotein A, apoprotein B, a bunch of apoproteins that dictate what they're going to become, you know, whether it's a triglyceride, a cholesterol, and the size of the cholesterol. And we'll speak about that in a later lecture in cardiology. Okay, in the next video, we're going to continue with this digestive and absorption process. And we're going to keep on going on to the remainder of the digestive process and essential vitamins and minerals that need to get absorbed, and mechanical stimulation of the colon, and how fiber plays a good role in the health of the GI. And after that, we'll begin with pathology.